Well, I think, in my opinion, that single-cell multi-omics are the future, but also the present. And I think that these novel techniques could give us more, more resolution for questions, biological questions of what are these tumor cells expressing and what make them different from another conditions. For example, we studied Waldenström, but also IgM and GUS patient, and also some healthy donors. And this could give us very, very powerful insight of what these cells are expressing. In our dataset, we observed that tumor cells from Waldenström, but clonotypic cells in IgM and GUS were expressing some shared markers, in example, the CD1C, but also they differ in some other markers. And we have to take a deep look into this analysis and these results, because maybe here we have the key to know which of these patients of IgM and GUS are going to progress to the Waldenström disease.